Hello everybody again. Second part of interaction analysis with Jörn. Ready?
Yeah.
Ready by year one?
Thanks a lot.
Thanks.
So I just want to say Nico made the program, so I'm sorry that you have to listen to me now twice in a row. So that's that's his. That was not my idea. All right. So we talked now a lot about this affinity purification mass spectrometry, about proximity labeling mass spectrometry. So that would always be, so the goal is that you enrich a complex a structure that you then want to study by mass spectrometry. An alternative approach to that, what gives you complementary data, is not to enrich your structure, but basically to follow your structure over a separation technique, right? And that is basically called this protein correlation profiling mass spectrometry. And usually what that is based on classically is on centrifugation approaches, right? When you look in classical biochemical cell biology textbooks, you always have this cell fractionation based by centrifugation. This has been done since 50 years, right? So where you have the differential centrifugation or the equilibrium density centrifugation, and dependent what kind of separation technique you use or combination of techniques, you can fractionate the components of the cell in order to study them, right? So you can, for example, have the soluble proteins. It's very easy, you know that you can spin down the nuclei, you can spin down mitochondria and enrich those, or chloroplasts, in order to study those. So there, in general, you would use a lysis buffer that does not contain detergent or only very, very mild detergent. You break the cell, you have this crude cell mixture, you perform these differential centrifugation approaches, and then you could study your protein or your organelle of interest. So you can do exactly the same and then use quantitative mass spectrometry to study the organelle proteome or to study also protein complexes. So how does that work? So you would have your density fractionation or your differential, basically pelleting fractionations, and then you take these fractions and then you analyze them by quantitative mass spectrometry, either using a label or using label-free approaches. And then, for example, if you do these step fractionations, you could look at your target organelle compared to your other organelles, or if you do these gradient density centrifugations, you can basically follow your organelle distribution over the entire gradient, right? I mean, this is a bit abstract, but I think it will get more clear when I show you respective data. So here we use protein correlation profiling to study the proteome of the autophagosome, right? So this is the same picture I already showed you in the last talk. So here we are interested in this double membrane vesicle. So for example, when you starve the cells, they form these double membrane vesicles as a response to starvation. It enwraps cytoplasm, and then this double membrane vesicle fuses with the lysosome and it gets degraded. We were interested in the proteome of this organelle of the autophagosome, and we used protein correlation profiling to study the proteome of the autophagosome. So this is now not classical protein-protein interactions, right? But it's more like organelle proteomics to ask what is inside the organelle. If you want to, it's a bit complementary to proximity labeling, no? You want to know the neighborhood of your protein and how this changes depending on the different stimuli. So, and we did this again in combination with SILAC. So this is a bit a busy slide, but I will walk you through this. So we have here our differently labeled cell cultures, right? So we have a heavy label and the light label. So, and then we would treat our cells by, now in this example, amino acid starvation. We treat the cells with amino acid starvation. This leads to the formation of the autophagosome. And then the autophagosome, we wanted to purify and to study by mass spectrometry. So how do you do an organelle purification? In this example, we use gradient centrifugation. So we did udixanol gradient, that's nothing different than a sucrose gradient. You have a sucrose gradient, and then you fractionate your organelle, and then you combine your different fractions based on the density of your gradient centrifugation. So now comes the trick. So here you keep your fractions. These would be the original fractions that you generated. And in this control experiment, you combine all the fractions again. This is like an internal standard. This is like all the proteins that are present in all of the fractions, right? And this internal standard, you spike now in a 1-to-1 ratio to your original fractions. This is one label, this is the other label, and now you can do relative quantification in single mass spec experiments. And this is an example how the data would look like. So here we collected 6 fractions, and when you do a Western blot, so LC32 is the marker protein of autophagosomes. So you do a Western blot, and then you see that autophagosomes peak in fraction 2 and 3, and then we have less, right, distribution over the gradient, and there's basically nothing in fraction 1. So this would be the Western blot readout, and this is the mass spec readout, right? So here in, what is that? Green, turquoise color, you have this original distribution over the gradient, and then in violet, this would be the standard, right? The average presence of all the proteins in all the samples. And because we have spiked that together, we can now measure fraction 1, we measure fraction 2, 3, and so forth, and then we can relatively quantify our protein of interest compared to our internal background, right? And this quantification is very accurate compared to any label-free quantification. So this works super well, you know, so here would be again the example, but there would be another protein, actin, which is also known to be in autophagosomes. You see the mass spec data, this would be the Western blot data, and this would be P62, another autophagosomal protein. So you can see that the mass spec and the Western blot, they always fit, right? So this works really well. But because this is not an enrichment approach, it's a fractionation approach, we don't just have our autophagosomal proteins. Of course, we also have contaminating other organelles which were in this fractionation. And here, for example, we have a mitochondrial protein, VDAC1. It's a protein of the outer mitochondrial membrane. And you see our mass spec data, and this would be the Western blot. Or we have nuclear proteins, so mitochondria and nuclei are quite dense. So in this density equilibrium centrifugation, they were at the bottom of the tube. And you can see also here our nuclear proteins, they have a completely different profile. So now the nice thing about mass spec is that you don't do Western blot and quantify one Western blot after the other, right? We generated thousands of protein profiles. So and then with these thousands of protein profiles, what do? You do a cluster analysis. So here we used fuzzy C-means clustering, but you can use K-means clustering, any basically clustering analysis you would also use for transcriptomics data, for example. And then we came up in this example with 3 clusters. And then cluster A we defined as autophagosomal cluster. So here we have all our known autophagosomal marker proteins plus many more. Cluster B would be ribosomes, Golgi, and cluster C would be ribosomes again, histones, exosomes, right? And now basically you have your organella cluster, and you can ask what kind of proteins that you did not expect, you know, that's why you did the study, are also in our autophagosomal cluster. And so the autophagosome is tricky because it's the garbage bag of the cell, right? So anything can be inside the autophagosome, right? But basically to try to make a bit sense out of the data, we did a Goterm analysis on all the proteins that were in our different clusters, right? Cluster A would be our autophagosomal cluster, cluster B and C would be our contaminating clusters. And for example, when you do this vesicle fractionations, you're always scared that you actually are not looking at autophagosomes, but at ER vesicles, right? Or that you look at Golgi-derived vesicles. And then, for example, when we look at the Goterm Golgi, you see that the Goterm Golgi peaks mainly in cluster B, right? The Goterm ER peaks in cluster B and in cluster C, right? So it seems, of course, this is a continuum, right? It's not a black and white separation, but it seems that, for example, Golgi ER are in cluster B and in cluster C, and in cluster A, basically, we have a lot of cytosol, a lot of cytoplasm, We have a lot of proteasome, which is a known cargo of the autophagosome. So it really seems that in cluster A, we have an enrichment of proteins which can be in the autophagosome. Then when you have this data, then the next question is, okay, how do you cross-validate that? What could be an experiment to show that this distribution and cluster makes sense? And for this, you could do a classical affinity purification. But here, we used cells that express GFP coupled to LC3. So this is our autophagosomal marker protein. So and then you can do an anti-GFP affinity purification without detergent. So when you open the cells without detergent, you leave all organelles intact. And then when you pull on GFP LC3, you do not just enrich GFP LC3, but you pull down the entire vesicle, the autophagosome. So it's basically an affinity purification on organelle level. And then you do this under control conditions and under different treatments for autophagosome, and then this would be the classical mass spec-based setup, right? And then you can compare the enrichment in your affinity purification based on your proteins when you distribute the proteins in the different clusters. And then you can see, again, because this is organella level, this is not black and white, but you can see that in the cluster A, these were basically the proteins that we defined as autophagosomal proteins. We have the highest enrichment in our anti-GFP affinity purification, and cluster B and C, we have a gradual decrease, right? So this would be at least some kind of validation experiment that this initial cluster distribution was sensible. And then you can continue with different biochemistry approaches. In this example, we did this specifically for the autophagosome, but there are many groups that do this on a cell level to get basically an organelle map of the entire cell. And I would like to show you some data which was not from our group, but this is the group of Catherine Lilly in Cambridge, I think she works. And she does this so-called LOPIT. This is localization of organelle proteins by isotope tagging. So it's an acronym, but basically it's again, she used instead of chemical, instead of metabolic labeling, she used this chemical tagging, and then basically does the same. You have here the classical gradient centrifugation, and then she used these chemical tags to then follow the distribution of proteins over the gradient. Or what she now came up with a new approach is where you don't use gradient centrifugation, but you use different step centrifugations. So you centrifuge with a different G-force, and then you pellet one organelle after the other, and then you study basically these different pellets, if you want to, right? And then she also gets these type of profiles, right? So this would be then either the profile over the gradient or the different pelleting steps, and then she can generate these very nice maps. I have another example here. This would be a principal component analysis of all the proteins, right? So each dot here is one protein, and you would have here the component 1 and component 2, and this would be with this step centrifugation, and this would be with the gradient centrifugation. And the color code corresponds to the different organelles, right? So then, I mean, maybe you know this data from single-cell RNA-seq data, right? They often present this in this principal component analysis. You can use then artificial intelligence or some computer algorithm where I'm not so familiar with to basically ask what kind of clusters do you have in these clouds. And you have your marker proteins, and then the hypothesis is that, for example, if we have here the mitochondrial cluster, right, you would ask, okay, which other proteins are close to my mitochondrial cluster and could be new proteins which play a role in mitochondria, maybe in my specific setting or something, right? So, and with this, you basically can then generate a network of the entire cell and try to understand the dynamics between the different organelles. It's one thing to look at the known organelles, but then of course what is super interesting if you have like compartments in between organelles, right? So there the hypothesis would be maybe these are organelle contact sites or proteins that play a role in organelle contact sites, and where you could try to go a bit more in detail in these type of analyses. Yeah, just to mention that. So then, this were example where you fractionate organelles. So for this organelle fractionation and organelle interactomics or organelle networks, usually you use centrifugation approaches. If you want to study protein complexes, you can do exactly the same, but you don't use fractionation, but you use size exclusion chromatography. So here we used, as an example, we studied BRAF protein complexes. BRAF is the most frequently mutated kinase in human cancer. It's a part of the MAP kinase signaling, right? And we were wondering what are the interaction partners of BRAF, and we used this size exclusion chromatography. Just to give you a very brief background on the biology, we have under wild type conditions, if you activate the growth factor, usually these are receptor tyrosine kinases in the plasma membrane, you activate RAS activates BRAF. This is like MAP kinase signaling, right? And then you have MEC-ERK goes into the nucleus and it signals proliferation or differentiation. If you have mutated BRAF, this mutated BRAF is not dependent anymore on any input signal. This is this V600E mutation. This locks the kinase in a constitutive active variant. And this constitutive active kinase constantly activates MEC-ERK and basically constantly signals proliferation and growth. And this is then how it comes to tumor growth. So in this project, we wanted to study the protein-protein interactions of wild-type BRAF and of our mutated V600E BRAF. And then we did basically exactly the same as in this gradient centrifugation setup, but we used size exclusion chromatography. So we have here our internal control, and then this was now a triple Silac experiment. We have here the V600E, so meaning the oncogenic BRAF, and we have wild-type BRAF. So then we enrich cytoplasm. We do a molecular weight cutoff filtration to get rid of all the small proteins and small complexes and only take everything which is larger than 100 kilodalton. And with this 100 kilodalton fraction, we perform size exclusion chromatography, right? So this would be a chromatography run where we generate 56 fractions. And we do three size exclusion chromatography runs. One for our internal control, one for the oncogenic BRAF, and one for the wild-type BRAF. So then we have our size exclusion fractions, and now we mix here the original size exclusion fractions, so fraction 1 with fraction 1, fraction 2 with fraction 2, and we have our 56 fractions. And now this here would be again our internal standard. Here we combine again all of the fractions, and then from this combined fractions, we spike into our original fractions in a 1:2 ratio to be able to do this relative quantification, similar how we did this with the centrifugation approach. So the data looks super nice. These are very heavy experiments on the mass spec side, right? Because in total, we generated 56 fractions. So these are basically 56 mass spec runs you have to do for one single experiment. So it's a lot of mass spec time. But the data looks very nice. So here we would have our fractions 1 to 20. And each line that you see here is the profile of one protein. And this would be now an example of the 60S ribosomal subunit and the 40S ribosomal subunit. You can see that they nicely elude here the 60S, and we have here the 40S ribosomal subunit. So this was just as a quality control that the experiment worked. But then you can do this now, because we did this on a proteome scale, right? You generate thousands of protein profiles, and then you can analyze these again by different clustering approaches in order to group all the profiles that have a similar pattern, right? And you can see here that we generated then many clusters. This would be a hierarchical clustering approach in this experiment. And you basically look at the clusters to study what kind of protein complexes you were able to separate. So I showed you the 60S and the 40S ribosomal subunit, but for example, we had a very nice cluster for the proteasome, cap, the core particle, and many different ones, right? So it's really a very robust, very reproducible approach. And then you can study the proteins you are interested in, right? In our example, we were interested in BRAF, and then we asked the question, which proteins differ the most in their elution profile, depending if we have the oncogenic version of the protein or the wild-type version of the protein, right? We wanted to study differential interactions between the oncogenic and the wild-type. So then we asked, okay, which proteins change most in their elution profile dependent on which BRAF variant is inside the cell. And this is this plot that would be basically the sum of the squared difference between the oncogenic and the wild-type version. And what you see here is that BRAF itself is always the protein which is most different in its elution pattern, dependent if it's in the oncogenic variant or if it's in the wild-type variant. And that was very interesting for us. So here we have, then when we looked at the profiles, you see this would be our 60 or 54 size exclusion fractions, and this would be the relative intensity. And we have here in dark blue the profile of the V600E BRAF isoform, and in not blue, green, sorry. In dark green, this V600E isoform, and here in transparent green, it's wild type. And might be a bit difficult to see, but we have here a dark green peak in fraction something, what is that, 20 something, and then another one here. And you see here in transparent, this will be the elution profile of the wild type variant. So basically what this tells us is that we have a large complex in the oncogenic variant, and this large complex is only very little abundant in the wild type variant, whereas in the wild type variant we have a small complex, and this small complex is much less abundant in the oncogenic variant. So we have two different complexes dependent on wild type or oncogenic BRAF. And the same as a validation step you can do then with blue native SDS page, right? So this would be a blue native exposure, and you can focus now just on this wild type and this V600E lane. And what you can see, these blue native pages always look ugly, right? It's always just like here, but it's under native conditions. You can see that here in the wild type variant, we have a small complex, which is very much abundant, and there's some smear here. And when we go to the V600E variant, you see we have here a large complex, and we have less of this little complex, right? And this is the stronger exposed ones, so you can also see this, right? So the same that we saw by mass spectrometry and size exclusion chromatography, we could recapitulate by a blue native page and also see these different variants. But again, with blue native page and Western blot, you look at one protein at a time, and you have to know to which proteins you want to look at. With mass spectrometry, you can ask, okay, which other proteins are there and have a similar profile to my protein of interest? For example, what we then saw is that we have CDC37. This is part of the HSP90 chaperone complex, which is important for kinase loading. So basically, CDC37 provides substrates for the kinase to be phosphorylated. It's a known interaction partner of BRAF. So you can see that the CDC37 nicely coelutes with the large complex, but it does not coelute with the small complex. On the other hand, we have 1433 proteins. 1433 proteins are important for for kinase priming, but also for the turn off of the kinase. And you can see that these 1433 proteins, they nicely bind to the small complex, but not to the large complex. So these were known interaction partners of BRAF. So it's known that BRAF interacts. But what was not known is that we have these different variants and that we have differential complexes. Because when you do an IP, you only get binary data, right? When you have an IP, you know my protein binds to X, but you don't know the stoichiometry of the complex, and you don't know the underlying makeup of the complex, right? You're basically blind to see that there are two different types of complexes. You have to use size exclusion chromatography or some other method that discriminates on size in order to discriminate these complexes. With an IP, you would never be able to see these differences. But then to prove the data, so once you have this size exclusion data, then you can perform a classical IP and ask, okay, what proteins do I find in my IP, right? And then we generated this network. So we have here BRAF in the center, and then the color code is that all the yellow proteins, we find stronger binding to wild-type BRAF, and all the blue proteins, we find stronger binding to the oncogenic BRAF. And what you can see here now is that CDC37, the protein that we know that co-elutes with the large complex, binds stronger to the oncogenic variant of BRAF, and the 1433 proteins, at least 2 of them, which co-elute with the small complex, bind stronger to the wild-type variant of BRAF. But we also found new proteins. We have a phosphatase subunit and isomerase, which we also found with differential interaction. And now with this IP data, we can go back to our size exclusion data and ask how are the complexes, how are the profiles of these proteins. You have to be aware of that the size exclusion data is very bioinformatically very complicated to analyze on a global scale. But when you have a affinity purification and you know for which proteins you are looking at, you can very easily take your targets and go back and ask, okay, how does the profile look like? So and then, for example, for this PP2A, which would be the phosphatase subunit, would bind stronger to the wild-type variant of the protein. We see that this co-elutes with the small complex, which is more abundant in the wild-type, and then this FKBP12, which binds more to the oncogenic variant, we see that it co-elutes with the large complex, so it binds stronger to the large complex, right? So you can really see that the affinity purification and the size exclusion data is very complementary, and you can learn something about the makeup of your different protein complexes. And then you can continue and validate all this by classical affinity purification. So here we purified BRAF itself, and we have wild type and oncogene, and you can see that the CDC37 binds stronger to the oncogene, and the 1433 binds stronger to the wild type. When you do the opposite P, you pull on CDC37, you see that the oncogene binds stronger to CDC37 compared to the wild type. So this is all consistent and makes sense. And then as a last piece of biological information, it is known how it is known how 1433 proteins bind to BRAF. We have 2 phosphorylation sites in BRAF. We have this serine 365 and serine 279. So these residues have to be phosphorylated in order for 1433 to bind, right? And what you can see then is that in the small complex, the low molecular weight complex, these phosphosites are present, and also 1433 is present. Whereas in the large complex, these phosphosites are not present, and also 1433 is not present, right? So you can really try then to describe or work out the molecular mechanism that is behind this different complex organization. So and then as a model, basically we saw that under wild type BRAF conditions, BRAF has to get activated by RAS, so we have these negative phosphorylation sites have to be removed in the long run, and we have this low molecular weight complex with less active. In BRAF V600E, we have the same complexes present, but we have more of this high molecular weight complex, and this high molecular weight complex is then characterized by CDC37 and HSP90, which is important for substrate loading. So we were able to discriminate the different complexes, and we could show that the larger complex is more active and is characteristic for the oncogene, whereas the smaller complex is less active, and this is characteristic for the wild type. As a last message, you can do exactly the same, not by size exclusion chromatography, but by blue native page, right? So you would run your blue native page, and this is called complexome profiling. You run your blue native page, and then you cut your blue native page in slices, and you measure the proteome or the proteomic composition in these different slices, and then you get exactly this as a size exclusion chromatogram. You can get basically a chromatogram over your blue native page separation of your different complexes. So this is it. I tried to be fast. Protein correlation profiling is very powerful if you want to study the dynamics of the proteome of an organelle or of protein complexes. You would couple that either to gradient centrifugation or to size exclusion chromatography. You can also use this native page if you want to. And I would always argue that it's a plus to do this in combination with the classical affinity purification or the proximity labeling, because it's a complementary data set, and the combination of both approaches helps you a lot in data interpretation and in studying the regulation of your proteins of interest. This would be it for the second part.
Many, many thanks. Oof, a lot of data, a lot of information from zero. Do we have any question? Thanks. I was thinking that. Questions? Oh.
Oh, very interesting talk. I have a few questions about the sample preparation for the protein correlation using size exclusion. Did you have to take any special care to prepare samples and make these reproducible for keep the different complexes bound? Yeah. And another small question is regarding the type of interaction you are recovering in the classification you show in the first slide.
Yeah. So for the size exclusion data, we found that this is very reproducible. I mean, there we basically opened the cells with a classical downser without any detergent in the sample, and then after you opened the cells, of course, we kept the ratio of buffer and cell pellet always constant, but then because you only do like this 2 centrifugation steps before you go on the sec column, this is really very reproducible. This is not necessarily the case for the gradient centrifugation. The gradient centrifugation, because dependent on how you collect the fractions of your gradient, if you collect them from the top, if you puncture them, and I mean there it's technically more challenging and you need more replicates. But for the sec data, this is really very robust. For the second question, that's a very good point. We never really addressed that. I mean, so for the interactions we found, I would assume that these are more the stable interactions that have a lower dissociation constant. We never tested differential mixing and see if we see any dynamics. Again, I think to really interpret the data, you have to couple it to an affinity purification and then to know which proteins you are looking for. Otherwise, just based on the profile, it might be difficult to interpret the data.
Hi. I have a question regarding the use of affinity purification to validate the PCP that you presented at the beginning. Is it necessary to have the bite anchored to the membrane of the organelle that you want to purify, or this is not a problem when the protein that you are using as a marker is inside the organelle? I mean, it's not like...
Yeah,
or should be in the surface.
If you want to use the affinity purification, it has to be surface accessible, right? Otherwise, this would not function. I mean, if you don't want to use mass spectrometry, you could use imaging, right? And then basically look for co-localization, right? By confocal microscopy, that would be a non-mass spec-based approach. But if you want to enrich, it has to be membrane. You have to be able to do an IP.
Okay, many thanks. Do we have one more extra questions, or we pass to the flash talks of today? Okay, many thanks, Jörn. We have Jörn on Saturday for proteomics.